In the end, human enhancement isn't just some fantasy anymore. It's something we might actually see in our lifetime. With all these new technologies growing so fast, we could soon have the chance to boost our bodies and minds way beyond what's natural. It sounds exciting, but also kind of scary. I mean, what happens if only some people can afford these upgrades? It's definitely something to think about. Whether we are fixing health issues or pushing our limits, the future of human enhancement is coming and it's going to change everything.
Well, how does it work, the PCR? Like we said before, the PCR is a technique that allows the DNA to be amplified, and it's made by this cycling process of cooling and heating. Well, the first stage is the denaturation. That is when the reaction of the mixture of DNA is here to separate the two strands of DNA and breaking the hydrogen bonds between the nitrogen bases. The second one, it follows the temperature and reduces to allow the primers to bind. And the last one is when the heat is repeated again and stop the tag polymerism, and this helps to synthesize new DNA strands.
The applications on real life of PCR are microbiology. It allows crucial observation of organism detection. Biology, it detects and characterizes the nucleic acids of viruses. Mycology and parasitology, they enable early identification of microorganisms. Dendricity, it has become a standard diagnosis and research tool.
PCR method had a lot of advantages nowadays. I investigated and there are four most important of them. The first one is that it's specific. Using PCR testing helps us to distinguish DNA sequences by just one nucleotide. The second one is that it's versatile. This technique can be used for many applications, like criminal investigations and paternity tests. The first one is that this method has a rapid results. PCR technology creates replicas that deliver the results of their investigations faster than other methods. The fourth one is that it's an efficient method. This method is very helpful when the DNA sample is limited, as it can amplify a small amount of DNA sample to 1 million of copies.
I'm Natalia, and I'm going to talk about the stages of PCR. Polymerase chain reaction, or PCR, is composed of 3 stages: denaturation, annealing, and extension, which every stage is repeated for 20 to 40 cycles. Denaturation is the stage when the double-stranded DNA template is heated approximately at 95 degrees to separate the strand into 2 single strands. Then the next stage is the annealing. The temperature is lowered about 50 degrees so the primers, which are the short sequence of DNA, can bind into the strand. For last, the temperature in the extension stage, the temperature is raised at about 72 degrees and the DNA polymerase extends the primers. This process is repeated multiple times to amplify the DNA target.
Advantages of this is the contamination. PCR is highly susceptible to contamination from various sources, including environmental DNA or RNA, which can lead to false and positive results. Innovation and errors. Innovators in the samples, such as heme from blood and samples, can disrupt the PCR reaction and reduce sensitivity. DNA polymerase can make errors during amplification, leading mutations or incorrect results. And the third one is cost and complexity. PCR requires an specialized equipment and reagents, making it a relatively expensive technique.